During this course, we will discuss about the theory as well as practical portion related to the vaccine designing. Do you have any idea about the vaccine designing? Anyone? Hmm? Okay, no issue. Abdul, Abdul,
Yes, ma'am.
can you please introduce yourself, your name, and your education background? That's it.
Ma'am, my name is Nawful Fazil Khan and I am pursuing B.Tech Biotechnology, currently in 5th semester from University University Bareilly.
You are from Bareilly.
No, ma'am, basically I am from Rampur, but I am pursuing B.Tech Biotechnology from Bareilly.
Which college?
Ma'am, Inverted University.
You are from Inverties. Okay.
Yes, ma'am.
Okay. Next, Afrin.
Yes, ma'am. My name is Afrin. I have done Masters of Science from VNSGU.
Okay. Aishwarya.
Yes, ma'am. I am Aishwarya. I am studying in BSc Biotechnology in Tirunelveli.
Okay. You have any idea about the drug vaccine designing? Angel. Anjali. Bhargavi. Anjali.
Ma'am.
Yes.
My name is Anjali Nahri. I have just completed my degree in subject biotechnology and pursued master's in biotechnology in DT University, which is in Germany.
Which university?
BTU University is Berlin Phoenixen University, which is located in Germany.
Okay. Bharti.
Yes, ma'am. I'm Bhargavi from Chennai. From Chennai, ma'am. BSc Cardiac Technology.
Okay. Chandan.
Hi, ma'am. I'm Chandran, ma'am. I'm doing my BSc degree in Chennai, ma'am.
Oh, you are also from Chennai. Gayatri.
Hi ma'am, myself Gayatri Devi. I am studying BSc Biotechnology in Tirunelveli.
Haridarshini.
Good evening, ma'am. I am Haridarshini. I am studying BSc Biotechnology final year at Sri Paramakalyani College, Alvar, Kuruchi.
Okay. Mufida.
Hi, ma'am. Can you hear me?
Yes.
Hello?
Yes, you are.
Yes, okay, my name is Mufida. I am studying BSc Microbiology, Kannur University, Kerala.
Okay. Pragya?
Ma'am, it's Pragyan, Pragyan.
Okay, Pragyan.
I'm actually studying B.Tech Biotechnology from Amity University, Noida. I'm in 7th semester.
You are from Noida?
Yes, ma'am. Amity University.
Amity University. Okay. Prashant?
Good evening, ma'am. I'm Prashant Kumar from Punjab. I'm currently studying in MSc in Clinical Microbiology from Lovely Professional University, Punjab.
Okay. Next, Poonam. Rahul. Hello.
Rahul Mahi from Kolhapur.
Okay. Sri Lakshmi.
Good evening, ma'am. I'm Sri Lakshmi. I'm doing my MSc in Molecular Biology at Yuvarajas College, University of Mysore.
Okay. Suma. Okay. You have anyone have any idea about the vaccine designing? What is the vaccine designing? What is vaccine? Anything? Sorry. Okay, no issue. Sorry, I can't. Okay. Poonam, no issue. Now you please tell me, you have any idea about the vaccine designing? What is the exactly vaccine designing, and what is actual में vaccine? Have any idea?
Yes, ma'am. Actually, about vaccine designing, I don't have any idea, but about basic concepts of immunology and vaccine, I have some sort of knowledge.
Please tell me.
Ma'am, vaccine is basically like a drug or a chemical, we can say, that is being prepared from inactivated pathogenic species. मतलब जिसको हम बॉडी के अंदर इंजेक्ट करते हैं जिससे कि इम्यून रेस्पॉन्स जनरेट हो सके, मतलब पैथोजेंस के, जिससे कि अगर फ्यूचर में अगर एक्टिवेट करें तो उसको मतलब मार सके वो.
Okay. Anyone? Amity University. Pragyan?
Yes, ma'am. So vaccine can be defined as any component of biological nature that involves producing an immune response. It can be a pre-exposure prophylaxis or post-exposure prophylaxis both. The vaccine technology also includes like studying the pathogenic compounds, the epitopes, what part of the pathogen creates an immune response, identifying it and creating it in lab so that an immune reaction is produced but not the disease.
Okay. Anyone other? Gayatri, Sri Lakshmi, Bhargavi.
Ma'am, a vaccine is a virulent form of the strains which we use to induce the immune response in the organisms, in the human's body.
Okay. Please, so you all have little bit idea about the vaccines, na? Or in vaccine designing? Any idea related for vaccine designing?
Ma'am, there are various methods for designing vaccines, ma'am.
Like?
Live attenuated.
These are the types of vaccines.
Yeah. Yes, ma'am.
I'm asking about the vaccine actually designing. What are the basic steps to design a vaccine? It is basically bioinformatics. Okay, no issue. In this 3 days workshop, we will discuss about the complete tools and techniques that are used to design a vaccine. Okay, so we start today's lecture and first we understand what is actually vaccine. Okay. What is vaccine? This vaccine. Let's suppose this is a person. In layman's language and very easy language, they understand what is the actual vaccine. This is a pathogen. Okay. Now this pathogen attacks this particular humans, animal, any plants, animal, or other. Okay. Now in this particular human, there are two types of immune system. First is the innate immune system. And second is the adaptive immune system. Okay. These are the two types. This innate immune system is by birth, and this adaptive immune system is during the death. Okay, let's suppose this is first, this particular pathogen pass through this innate immune system. Whenever it will pass through this immune system, then it will activate the adaptive immune system. Okay, if adaptive immune system will activate, then it will form, first it will form T cells. Then T cells basically T cells, T helper cells, T cytotoxic cells, T suppression cells, or different types of cells. Okay. Whenever this T cells will activate, then it will activate the B cells. Okay. Then it will activate the B cells. Okay. During this type, there are two types of B cells. First is a immunoglobulin, and second is a memory cells. Okay. And first is the immunoglobulin. Immunoglobulin like A, M, D, E, G. This immunoglobulin cell or antibiotic, this is also known as antibiotic, this will activate and kill this particular pathogen. This is the second thing. Now, this memory cell will also be made. What is this actually memory cell? This memory cell is for the future or for the second response. Okay. And this is our immune system. This is the exact working of our immune system. Okay. But in case of vaccine, we generate the immunoglobulin, this antibiotic. Okay. But in this case, we use the same bacteria or same viruses. But this bacteria or this viruses is not in its actual position. It may be attenuated, it may be live attenuated, it may be killed, it may be any small portion of this antibiotic and of this particular bacteria. Okay, this one. Okay, this is the vaccine. In this vaccine is a biological preparation that train the immune system to recognize and fight against the specific pathogen, but without causing the actual disease. Okay. Whenever this pathogen will activate, then it will cause the disease. Okay. But in vaccine, we use this bacteria. This bacteria is in inactive form. That's why it will not cause any types of infection or any types of pathogenicity. But the function is same. It will pass the innate immune system, then it will activate the adaptive immune system, then it will activate the T cells, B cells, immunoglobulin cells will form, memory cells will form, and this antibiotic cells will kill this inactivate viruses. Okay, this is the vaccine. What is a vaccine? Vaccine is a biological preparation that train the immune system to recognize and fight against the pathogen. Okay. During this process, this memory cells plays a most important role. Okay, memory cells is for the secondary response. Understood? Now,
Yes,
this is the
ma'am.
okay. This is the actual working of vaccine. First, antigen introduction, immunoactivation, antibiotic production, memory formation, secondary response, and this what we already discussed. Okay, antibiotic introduction means vaccine introduce a antigen. And this antigen is weak, killed pathogen. It may be protein, it may be genetic. Immune system activation means it will activate the immune cells like dendritic cells, macrophage cells, antigen, and present T cells or B cells. Antibody produced from the B cells. Okay, memory cells like T and B cells that will create the long-term protection. Okay. Secondary response, if the real pathogen infect later, then this immune system is faster and strong, prevent the disease. This is the main purpose of vaccine. What is the main purpose of vaccine? The main purpose of vaccine is that whenever the real pathogen infect the human body or infect, then this memory cells will activate and prevent the diseases. Okay, this is the vaccine and this is the work of vaccine. Okay, now these are the different types of vaccine, like live and attenuated vaccine. In this vaccine, only use the live viruses or live pathogen. Okay, but weak. Weak means it will divide, but does not cause any types of pathogenicity. And it will strong and long-lasting immunity, and this is the MMR, BCG, and oral. Okay. Next is the inactivated killed vaccine. In this vaccine, we use the killed pathogen. Pathogen create the immune response. Okay, this is safe but require the booster doses. And the example is inactivated polio vaccine, rabies, and hepatitis. Next is the subunit and recombinant polysaccharide. In this types of vaccine, we use the some part of pathogens like protein, like sugar, like capsule, like nucleic acid, etc. Okay, but it also have some few side effects. And the example is the hepatitis B, HPV, and pneumonal vaccine. Okay. Next is a toxoid vaccine. Toxoid vaccine means we will take the inactivated toxins. Inactivated toxin, not a pathogen. Okay. It will protect against the toxin producing viruses like bacteria and yeast. mRNA vaccine. In mRNA vaccine, we use the mRNA. COVID-19 vaccine like Pfizer, BioNTech, and Moderna is based on the concept of mRNA. Okay. Next is a vector-based vaccine designing. In this vaccine designing, we use the harmless virus as a delivery method. Okay, for the pathogen genes. Okay, and the example is Oxford AstraZeneca COVID-19 vaccine and Johnson & Johnson. Okay, this is the different types of vaccine: live attenuated vaccine, inactivated vaccine, subunit recombinant polysaccharide vaccine, toxoid vaccine, mRNA vaccine, and vector-based vaccine. Okay, and this is the summary. Live, inactivated, subunit, polysaccharide, conjugated, toxoid, mRNA, and viral vector. Clear? Clear?
Yes, ma'am.
Question?
Ma'am, I have a doubt. Can I?
Yes.
Ma'am, if the pathogen is killed, how can they activate the immune system?
Yes. Anyway, this is our immune. Our immune system recognize the foreign particle. Okay. This foreign particle is activate or inactivate. This will not recognized by the immune system. Okay. Understood?
Okay, ma'am. Clear. Yes, ma'am.
Now clear?
Yes, ma'am.
Okay. Now, these are the basic steps during the vaccine design. First, sequence retrieval and structural analysis. Okay. Sequence retrieval and structural analysis. Epitope prediction. Epitopes. Vaccine constructions. Secondary tertiary structures and validation. Molecular dynamics and expression analysis. Okay. These are the important 5 steps during the vaccine design. Understood? Now, first step is a sequence retrieval. Sequence retrieval means we retrieve the sequences. For sequence retrieval, we use a database like NCBI, like UniProt kit. For protein structural retrieval, we retrieve the protein sequences or genome sequences. For this, we use a database like NCBI, like UniProt. Next is the structural analysis. Structural analysis means we check the physiochemical properties, secondary structure, disulfide bonding, antigenicity, allergenicity, and tertiary structures. Okay. Now these are for the epitope prediction. Means we predict the B cells epitopes, T cells epitopes. Okay. These are the basic component of vaccine: adjuvants, linker, 6 histidine tags, etc. And we again check the allergenicity, antigenicity, and toxicity. Like Vexigene, Allotope, Toxicity. Then this is for the assembly करते हैं, and then we assemble the vaccine. Then finally check the solubility and physiochemical analysis. This is the way by which we easily construct the vaccine. First we add adjuvants required, then linker, B cells epitope, again linker, MSC class 1 epitopes, then linker, then MSC class 2 epitope, and histidine. This is the complete setup of vaccine. This is for checking and expression analysis. For checking and expression analysis, we use docking. For protein-protein docking, we use Clespro 2.0 and PASDoc. For molecular dynamics simulation, we use iBot. For vaccine expression, Inverse, ZCAT, and SnapGene, and Benchling also a software. Okay. And this is for the immune simulations. Okay. These are the tools and techniques which we use during the vaccine construction. Clear? 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 Okay. Okay, so for NCBI, what is the NCBI? NCBI is a database. Okay, this is not under the DBT. Okay, NCBI is a software from the Zenbank. Okay, in NCBI is a big database. In this database, we know the information about the chemicals, about the literature, about the books, about the molecular, about the sequences, about the structure. Lots of databases are available in NCBI. Okay. For NCBI in Google, you simply search NCBI. Open this one. Okay. And open this first link. And now this is the homepage of NCBI. Okay. In this NCBI, lots of databases are available. Suppose you want to retrieve the protein sequences. So for protein sequences, you simply first, for nucleotide sequences, you simply select in all databases nucleotide, then your target sequences. And our target sequences is this one. Our target sequences is this one. MT520466. Okay, this one. MT520466. Okay, this is basically, what is this? This is the accession number. This is the accession number of SARS coronaviruses. Okay. Okay. Now this is the GenBank format. This is the GenBank format of this protein. Now this is the ORF1 polypeptide protein sequences, or we want the membrane protein. Okay, this is the ORF1 polypeptide protein. Framesip. This is the surface glycoprotein. We want the membrane protein because we want to design the vaccine against this one. Okay, so I simply click on this protein ID. Whenever we click on this protein ID, then we go or we switch on the protein databases. Okay, now this is the membrane protein from the COVID-19 protein. COVID-19, this is the number of amino acids, and this is the sequences. Okay, now I convert the sequences in FASTA format, so I simply click on FASTA. Okay, now this is the FASTA format, and first step is completed. Clear? Any doubt? Okay, so next step is, first step is clear. Second step is the structural analysis. For structural analysis, we use a software like physiochemical analysis. First, we check the physiochemical analysis. For physiochemical analysis, we use a software like Expressi Properum. So for this, in Google, you simply open Expressi Properum. Open this software, then open this first link, then paste the sequences. Here you paste the sequences not in FASTA format, only protein sequences. Okay, this is the complete FASTA format, so I simply copy only these sequences. So I copy the sequences, open this one and paste and compute parameters. Okay, now this is the information. Number of amino acids, their pi value, their molecular weight, their amino acid composition, total number of negative residues, total number of positive residues, number of carbon, hydrogen, nitrogen, oxygen, sulfur, formula. Complete physicochemical information are available on this page. Understood? What is this ProTparam? ProTparam is a software to check the physicochemical analysis of protein. Understood? Pragya, Rahul, Anjali, Abdul, Sri Lakshmi. Sorry, Pragyan.
Yes, ma'am.
Okay.
Yes, ma'am.
Okay, Pragyan. Okay, Poonam. Now, first step. Now, this first step is completed now. Physicochemical analysis. Now, secondary structure. For predicting the secondary structure, there are a number of softwares are available, like Chow-Fasman, like Gore method, like PSI-PRED, etc. Okay, so for finding the secondary structure of protein, we use a software like Chow-Fasman. For Chow-Fasman, we simply search Chow-Fasman. Okay, then they open this first link. That is the Chow-Fasman secondary structure prediction server. Open this software, then paste the sequences in FASTA format. So I simply copy this whole sequences. Okay, and paste and predict. Now this is your secondary structure prediction result. Okay. Blue, this red line indicate the helix, green sheets, blue turn, and yellow coil. Means these are the amino acids which are participate during in coil formation. These are the participate which are participate in helix formation, in seeds, like this. Okay. Total number of helix is 156. Total number of seeds 174. Total number of 10 is 19. Percentage 70%, 78%, or 8%. Secondary structure prediction clear?
Yes, ma'am.
Okay.
Yes, ma'am.
Okay. Now,
Ma'am, can you repeat this once again, please?
Prashant, this is a software that are used to find out the secondary structures of your protein. Okay. For this, in Google, you simply search Chow-Fasman. Okay. Then open this home page of Chow-Fasman. Then in home page, you simply paste the sequences in FASTA format. Okay. And this is the FASTA format of your sequences. So simply copy the sequences and paste, or whenever you paste the sequences, then you get this result, and this is the complete result. This yellow line, this red line indicates the helix, this green line indicates the sheets, and this blue line indicates the turn, and this yellow line indicates the coil. And these are the amino acids which are participate in coil formation, for helix formation, sheets formation, etc. And this is the complete number of helix 177, number of sheets 174, one number of turn 19, and percentage 73%, 78%, and 8%. Now clear, Prashant?
Ma'am, a yellow line is for what?
Yellow line is for the coil. This one. Coil, turn,
Okay.
sheet, helix. Clear, Prashant?
Okay, ma'am.
Okay. देखो. Secondary structure prediction and physicochemical analysis. Next is a disulfide bonding prediction. So this in Google, you simply search a tool like a Dyna 1.1 web server. Okay. Then open this software. Okay. This is the home page of this Dyna 1.1 web server. Here you paste the sequences in faster format. Okay, faster format means complete sequence or only these sequences? Tell me. Complete sequences or only sequences?
Complete sequence.
Complete
Complete
sequences.
sequence.
So I simply copy the sequences and paste. Then click here. Okay. Now this is your result. Okay. These are the bonds. These are the amino acid and their scoring values. Scoring values, bond, distance, and cysteine sequence position. Understood? By the help of this software, you easily understand the disulfide bond. Okay, next software is for to check the allergenicity or antigenicity. For antigenicity, to check the antigenicity, it means your compound is antigen or not. So simply open Bexygen 2.2. Open this first link. Wait. Okay, now here you paste the only protein sequences, not a sequences in faster format. So simply copy the complete sequence or only sequences. Complete sequences, only sequences, copy the sequences and paste and choose the target sequences bacteria, tumor or viruses is tumor and submit. Now your protein is antigenic, contain the antigenicity or non-antigen? It will antigen means it will have a capability to activate our immune system. Understood? If this threshold value is more than 5, more than 0.4, then it will indicate your protein is antigen. Okay? If this value is less than 0.4, then it will indicate your protein is non-antigen. Clear?
Yes, ma'am.
Okay. Now, another software to check the allergicity. It means this particular protein contain the allergenic property or not. For this, we use a software like Allotop. Allotop 2.4. Okay, then simply search and open. For this phase, first you log in yourself or register yourself, then perform the task. Okay, and I am already registered on this page. Okay, so here you simply paste the protein sequences. Okay, so simply paste. Okay, and submit. Now this is your protein. Now this is your final result. Your protein is allergenic or non-allergenic?
Non-allergenic.
Means your protein cause the antigenicity in your human body or does not cause any types of allergy. Okay? Okay. And tertiary structure. Okay? To check the tertiary structure of this particular protein, we use a software that is T-Rostra. T-Rostra. Okay, this T-Rostra, open this, so simply search the T-Rostra and open this link and paste the sequences in faster format. So maybe only paste the sequences, not a faster format. So simply paste the sequences, and here the most important point is you simply give your email ID, because this result is on your email ID. Then simply check, 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 check and submit. Okay, and wait. Now your result is submitted, and this and the result of your protein is mailed on your mail ID. Understood? And in this way, this second step, that is the structure analysis, is complete, and the result of this tertiary structure we will discuss in next class. Okay, now tell me, physiochemical protein sequencing, what is the vaccine, what are the steps of vaccine, protein sequence structure, physiochemical properties, antigenicity, allergenicity, disulfide bond, all tools and techniques clear? Any doubt, any question? Any doubt, any question? Sri Lakshmi, Abdul, Anjali, Hari, Prashant, Pragyan, Rahul, Mufida, Gayatri, Prashant, Sri Lakshmi, Suma.
Excuse me, ma'am.
Yes.
Ma'am, I want one favor from your side.
Yes, please tell me.
मैम आपने जो मतलब सारे सॉफ्टवेयर एक्सप्लेन किए मैम, लाइक स्टेप बाय स्टेप NCBI XKG, मैम could you please provide a मतलब समराइज मैनर इन अ सीरियल वे कि मतलब कैसे कैसे मतलब एक के बाद हमें यूज करना है एक ब्रीफ मैनर में.
हाँ, I will share the PPT and also recording of this lecture. Okay. Yes, Anjali, I will share the PPT. Okay, I will share the PPT. Okay, Abdul. Afreen.
Yes, ma'am. Thank you.
Welcome. Afreen.
Yes, ma'am.
Any doubt?
No, ma'am.
Clear?
Yes.
Okay. So, and next step is for the epitope prediction. What is the epitopes and what are the steps we use the epitope prediction? We will discuss in next session. Okay? Okay?
Yes, ma'am.
Okay, ma'am.
Okay. Okay then, thank you class.